Lipid metabolism, digestion, and absorption. This slide presentation reviews the process of lipid digestion and absorption with a focus on triacylglycerol molecules and cholesterol. The primary enzyme used in the digestion of lipids is lipase. Lipase cleaves the ester bond joining the fatty acids to the glycerol backbone. You can see here the glycerol backbone, and we have the fatty acids. Each fatty acid is joined to the glycerol backbone by an ester bond, and lipase is involved in cleaving the ester bond. In humans, lingual and gastric lipase contribute to little lipid digestion. However, with the activity of lingual and gastric lipase, some triacylglycerols are converted to diacylglycerols in the area of the mouth and stomach. Lingual and gastric lipase particularly cleave the ester bond of the triacylglycerol in the third position, freeing the fatty acid. Next, after the activity in the mouth and the stomach, the diacylglycerol molecules, triacylglycerol molecules, cholesterol, cholesterol esters, and phospholipids move from the stomach to the small intestine. There are two phases of activity in the small intestine. The first phase of activity involves emulsification of large fat globules by bile acids. This activity forms smaller fat globules called micelles. The micelle formation allows for further digestive activity by pancreatic lipase. However, you can see that colipase is required to activate the pancreatic lipase before it can have its effect. Pancreatic lipase acts on the triacylglycerol and diacylglycerol molecules, and it cleaves the ester bond linking the fatty acids to the glycerol core. And this activity of pancreatic lipase cleaves the ester bonds particularly at position 1 and position 3 on the glycerol backbone. This yields monoacylglycerol molecules and free fatty acids, which are then available for absorption into the small intestine. This diagram goes over the activity of lipid digestion at the mouth, the stomach, and small intestine, including the first phase of emulsification and the second phase involving enzymatic digestion by pancreatic lipase. We can see that lingual and gastric lipase cleave the ester bond joining the fatty acid to the glycerol backbone at this third position, as mentioned previously. We can see it in the mouth and in the stomach. And then in the small intestine, we have the first phase involving emulsification by bile acids and formation of the smaller lipid globules called micelles. The second phase is the enzymatic digestion phase, and with the enzymatic digestion by activated pancreatic lipase, we have cleaving of the ester bond at the first and the third sites on the glycerol backbone. The primary end products of lipid digestion are the monoacylglycerols and fatty acids. You can see here that the micelles and their digestive products, monoacylglycerols and free fatty acids, interact at the brush border of the small intestine and diffuse into the enterocytes. Once absorbed, short-chain fatty acids are transported out of the enterocyte into portal circulation. In contrast, in the endoplasmic reticulum of the enterocytes, monoacylglycerols, diacylglycerols, and free fatty acids combine to form triacylglycerols. The triacylglycerols, along with other lipid components such as cholesterol, phospholipids, and fat-soluble vitamins, are incorporated into chylomicrons. The chylomicrons exit the enterocyte and move into lymphatic circulation. This diagram provides a visual of the process of triacylglycerol digestion, absorption, chylomicron formation, and movement into lymphatic circulation. You can see that in the small intestine, large lipid globules are emulsified by bile, forming smaller micelles. These smaller micelles are then further exposed to digestive enzymes such as pancreatic lipase. The activity of the pancreatic lipase yields monoacylglycerol and free fatty acids, which can then enter through the brush border membrane into the enterocyte. In the endoplasmic reticulum of the enterocyte, these lipid components are reincorporated into triacylglycerols and integrated into chylomicrons with other lipid particles. Digestion and absorption of cholesterol is similar to triacylglycerols. Cholesterol esters are hydrolyzed via the enzyme cholesterol esterase, freeing the cholesterol. Phospholipids are also hydrolyzed. Approximately 1/2 of cholesterol consumed is absorbed, and the remaining cholesterol is excreted in feces. You can see in this diagram the absorption and excretion of the cholesterol from bile and from dietary sources. Here we see the cholesterol coming from bile combining with dietary cholesterol, and of this, 50% is absorbed and about 50% is then excreted in the feces. We will next discuss chylomicrons and their subsequent products, the lipoproteins, which are involved in the transport and delivery of lipid components through circulation and to various tissues. Again, the chylomicrons formed in the enterocyte are triacylglycerol-rich lipid particles, also containing cholesterol esters, phospholipids, and fat-soluble vitamins. Chylomicrons in the lipoproteins also contain specific apolipoprotein components. After the chylomicrons circulate through the lymphatic system, they are picked up by the liver, where triacylglycerol, cholesterol, phospholipids, and protein are packaged to form VLDL and HDL. So those are the two lipoproteins synthesized in the liver, VLDL and HDL. You can see that the liver is also responsible for synthesizing bile acids and new lipids. Here are the lipoproteins. VLDL produced in the liver is converted to IDL and then LDL as it releases lipid components when traveling through circulation. This diagram specifies the primary lipid components of each lipoprotein. Chylomicrons, as we mentioned previously, contain primarily triacylglycerol, particularly triacylglycerol from the diet. We can go down then to LDLs. LDLs contain primarily cholesterol and have the highest cholesterol content of all of the lipoproteins. HDL contains mostly protein as well as phospholipid. After eating, adipocytes absorb triacylglycerols and cholesterol from circulating chylomicrons with the hormone lipoprotein lipase. This allows for the storage of triacylglycerols in fat cells. Enzymes facilitating this process of triacylglycerol storage are active in the fed state. Adipocytes can also metabolize glucose to acetyl-CoA, which can be converted to longer chain fatty acids and stored as triacylglycerols.